Uh, Hello, everyone. I'm Isao Kashima, the instructor for molecular biology in our daily life course. Thank you for watching the video guidance. The administrative details listed here are available on the course website. If you have a device capable of scanning QR code, please check here or use hyperlink in the YouTube video description. If you have any question, please contact me by email. In this video guidance, I will cover three main topics: program highlights, and message from the first student to you, and a brief self-introduction. First, let me introduce aspects of this course that must be more detailed in the syllabus. The essence of this course can be summed up in the two words: research experience. By the end of this course, you will acquire basic molecular biology skills and critical thinking through individual project. In A part, we will conduct molecular biology experiment such as tuna species identification, as you can see here. In B part, based on skills from the A part, you will tackle individual projects like DNA barcoding or optimization of the PCR protocol for direct PCR. What comes to mind when you think of research experience? How can you learn about lab life before you join? Maybe reading well-known PhD comic could be one way. When I was postdoc, I often read comics with my colleagues and learned a lot of things while I'm laughing. Anyway, this course incorporates fundamental training in experimental science. Therefore, I explain you how to write experiment note, lab note, and their importance. All previous students' lab notes have been stored in either paper or electronic form, serving as variable archive. In addition, during the lab period, you will assign your own workspace and the bench space, as you can see here. And you may learn about importance of cleaning up the bench also. This is because you will conduct a lot of the experiment. And you will have also a lot of the discussion with colleagues. In the B part, you will work on your own project with flexible schedule at your own pace. Like this photo, everyone will do different tasks simultaneously, progressing together in the lab. Presentation will be conducted in the lab meeting style on the final day, presenting raw data and lab note. In this course, there is no need to extensively edit presenting material beforehand. The aim is to learn not for winning presentation, but for a shared presentation. This presentation is by Peak students. In fact, we enjoy his presentation, the insight he provided. This is received stamped, you can see here, of approval for unique and insightful idea. The mastery of fundamental technique in the first half is essential for enjoying the individual projects in the second half. Participants unfamiliar with the research will receive personal training to acquire skill. Moreover, throughout the course, the lab atmosphere will remain relaxed. Individual documents play a crucial role in the later half personal project. This time, we're going to use Google Docs. As you can see here, you will share this document with me, updating as you need it, and I will immediately review it and add comments. What's the essence of research? In this course, I hope you will learn the joy of setting your own timer and advancing your project rather than working in groups. This course design reflects how seriously and thoughtfully you consider your own results, rather than how much you knew in detail in advance. This course will be held at EDL Lab at third floor of Commerce East Building on Komaba campus. You can experience and consult with me before the lab session by booking a PreLab session while I'm in the lab. Please refer to the course website for detail. The main elements of research experience and lab experience in this course are indicated here as I mentioned. Okay, we move to the next. I will introduce you to introductory message from Pacific student about this course. First one, this course is gateway to molecular biology. No special skill are required. It means that if you are familiar with the molecular biology jargon, don't worry, we will support you and you can do self-study during the course. He was the first PIC student I accepted. In fact, he developed very interesting, unique protocol. Next comment. This sentence contains an important insight for both me and you. This way of thought to know why protocol work developed during the course is extremely useful to our students. As too often in an experiment, we just blindly following the procedure without learning the mechanism behind. In addition, learning while enjoying oneself is essential. This is a very well-written lab note. Excellent. Integrating course highlight and insight from the first student, the learning outcome of this course can be expressed like this. Through the concurrent progression of the theory and practice in the lab experience, you will experience a better reconnection of knowledge. This may prompt you to reconsider what it means to acquire knowledge. From my perspective, these experiences are actions where I can check that I'm not just robot in life science experiments, realizing the reason and the courage behind them. I've been talking a lot so far. Some of you might be wondering if I'm all right. Let me give a brief self-introduction not included in the syllabus. Certainly, Google has a significant focus on me. I'm on the Google Maps. The hat I'm wearing now This one? This is it? Since this photo is taken very well, I will request image without mosaic next time I meet Google engineer. Apart from this photo, I will still on Google Maps somewhere else in the world. If you find me, please get in touch with me. I have some reward ready. This is my CV. I have been researching life science, particularly RNA. To be more specific, post-transcriptional gene regulation in eukaryote cell. I trained at Max Planck Institute in Germany during my postdoc. At my time, my Max Planck Institute strongly influenced my enjoyable experience in international environment. Many tips and academic life introduced in this course were realizing during this period. In 2015, I shifted from the basic research to education. As I mentioned earlier, I currently working on the teaching and developing material to promote reconnecting of knowledge, especially in life science, here at Komaba campus. Today, we confirmed the contents not listed in the syllabus regarding the highlights of this course. We also understood the critical point of this course from the student perspective in the introductory text. Finally, I introduce myself. The administrative details of this course are listed here are available on the course website. If you have any question, please contact me by email. With that, I would like to conclude today's video guidance. Thank you very much for watching so far. I look forward to meeting you during the PreLab or actual course period. Thank you very much. Bye.